Evolutionary biology is arguably in its most exciting time ever. And Harvard University, which has been studying evolution for many years, has many excellent programs and individuals who are at the forefront of evolutionary research.
On to the next stage.
What motivates me as a paleontologist is really understanding sort of the history of life. What fascinates me is the fact that life and everything changes. So how has it changed? That's the empirical issue that the fossil record can answer. And what are the rules by which it changes? How much can it change? How fast? What's the quality of change that's possible? And that's what motivates my more theoretical questions that come off that fossil record. Nowadays, particularly with the capacity to use computer simulation, we can actually now rerun the history of life in the computer and see if any generalities emerge. One of the generalities that I think emerges is that some of the large-scale patterns of the history of life are actually, in principle, repeatable if we were to replay the history of life over and over again. The computer simulation, by using just 6 developmental rules and 4 selective pressures, is able to mimic the gross evolution of plants. It strongly suggests that if we were to replay the history of life of plants over and over again, we would get a similar pattern every time. So one of my major theoretical interests is the cause of the Cambrian explosion. That is the sudden appearance of almost all of the major animal body plans about 540, 530 million years ago. The cause of the explosion has been extremely problematic. There must be new genes involved, the environment must have been correct. Steve Gould, in his book Wonderful Life, emphasizes the role of chance in that. For me, I think there's a much greater degree of inevitability to the explosion once it starts. And so applying the plant simulations, the argument is that the same sort of thing that causes those plants to diversify from the 1 or 2 optimal solutions to 20 optimal solutions is exactly what happened in the Cambrian explosion.
We're studying in my lab the relationships among the different animal phyla that originated and diversified about 500 million years ago. And we do from traditional study of their morphology and their anatomy to the more modern studies of their DNA. One of the things that we do here is to study also the biogeography, which is trying to put together the distributions of the organisms with their evolutionary relationships and see where these organisms are found nowadays, whether this is the result of a historical process of, for example, the continents breaking up into other landmasses. That's what we call historical biogeography. So we overlap these 2 types of studies, the distribution studies with the phylogenetic studies, to come up with explanations for the current distributions of the organisms. One of our favorite animals for studying biogeography are daddy longlegs or harvestmen. This is one group of arachnids in the order Opiliones. And the reason why we have been focusing on these animals is because in general they are poor dispersers, which means that they don't cross oceanic barriers very easily, and therefore they are very loyal to the land masses where they have diversified. So by tracing back in time their distributions of those land masses and mapping that to the phylogenies of the organisms, we've been able to trace a lot of these biogeographic relationships.
My research is focused on studying growth processes in fossil humans as well as living apes and humans. I'm specifically focused on teeth because they contain a very precise record of development, which begins before birth and continues throughout our childhood until we become mature. So what's actually locked in the teeth is a very, very intimate history of an individual, and it allows us to get at growth and development in ways that we can't from other fossil remains. One of the most exciting new breakthroughs in the study of dental tissues is the use of something called a synchrotron, which is a very high-energy X-ray source that allows us to see inside structures without having to break or cut them. So this video shows this fossil from Israel imaged with synchrotron light, and you can see the teeth coming out virtually from the fossil bone. What we did is we used a special optic to see the internal development of each one of the teeth. Here you can see the canines, the incisors, and then we're actually able to virtually take the teeth out of the fossil jawbone and see exactly how much root has been formed, what stage the individual tooth is at. And then we can use an even better optic to see very fine details on the outside of the teeth. as well as inside the teeth. We can recognize stress. And then here we've used an even finer optic to see the fundamental structure of tooth enamel. What we're able to do is actually penetrate through these enamel prisms and recognize daily lines, or these 24-hour rhythms of tooth formation. So by using this technology, we can work at the microstructural level and we can work at the macrostructural level to see how this fossil juvenile grew and developed, whether it shared a childhood like our own or something more similar to earlier fossil humans.
Thanks to molecular biology, we have tremendous and increasing understanding of the genes and how genes are turned off and on and regulated that underlie the processes, the mechanisms that give rise to evolutionary change. This kind of information was beyond the dreams of evolutionary biologists more than 25 years ago, and it's bringing us tantalizingly close to the ultimate answers to a lot of fundamental questions in evolution that people have been asking for more than 150 years.